Hello everyone. Today we will be solving some questions related to the topic molecular biology, and the question came in the year 2015 in XLI, that is biochemistry, in question number. Then the question number is 18. What is the question stating? The question states that a 1.2 kilobase pair DNA fragment was used as a template for PCR amplification using primers P1, P2, P3, and P4 as shown in the scheme below. The annealing positions of primers on the template are indicated by numbers. Primers P2 and P3 contain single base mismatches as indicated by filled triangles. PCR was performed using primer pair P1 and P3 in one vial and P2 and P4 in another vial. The purified PCR products from the two vials were mixed and subjected to another round of PCR with primers P1 and P4. The final PCR product will correspond to a. So for solving this question, one has to know the basics about PCR, that is the polymerase chain reaction, and also something which is known as this single base mismatches, how these single base mismatches are being made by site directed mutagenesis. So that also I will be discussing before solving this question. So now first we shall look into that what is a polymerase chain reaction. Polymerase chain reaction, if you see, it was discovered by Carrie B. Mullis in 1985, and it has revolutionized the field of biotechnology and biomedical engineering and biomedical research a lot. So now there are different applications of this polymerase chain reaction, but how this has been developed, if you see that similar to this, something actually happens in our body. That is this replication. So in replication, you have actually the DNA which actually contains your encoded information, the genetic information in the DNA, and that DNA will be converted or will be actually replicated into another daughter DNA. Okay, so how this replication occurs in our cell? In the replication occurs by following three basic steps. So first one is unzipping, that is done by an enzyme in our body, that is helicase. So first your double-stranded DNA should be unzipped, or it should be unwinded to single strands. Okay, so that is done in our body or in our cell by helicase. Then there will be complementary base pairing, that is new DNTPs will come and form a complementary strand for to each of the parental strand, and there will be also a new sugar phosphate backbone will be formed as a result of the new daughter DNAs will be formed in this process of replication. So this replication process is actually mimicked in the polymerase chain reaction, where in polymerase chain reaction you actually get your product in a huge amount, in millions of quantities, that is in millions of amount you get your target DNA in polymerase chain reaction. So how it is been done? It has been done by the finding of a thermostable Taq polymerase. So in PCR, it's different from the replication in the way that in PCR we actually use high heat or heat denaturation to unwind the DNA instead of the helicase enzyme. So but there is a problem with that, because if you use normal DNA polymerase, which is actually responsible for the replication process in our cell, that is the DNA dependent DNA polymerase, then that enzyme will be unstable in this high temperature and will degrade instantly at 95 degrees Celsius. But in PCR, we actually unwind the DNA with the help of temperature. Okay, so we need something which can actually withstand this high temperature of 95 degrees Celsius. So scientists have found an enzyme known as the stack polymerase, which is actually taken out or derived from a Thermus aquaticus. Okay, from a bacteria known as Thermus aquaticus. So this stack polymerase can actually withstand very high temperatures like 95 degrees Celsius, and they can actually give a optimum polymerization activity from 70 to 75 degrees Celsius. So other than this, what are the things that is required for this PCR to happen? So you need a template DNA, as you also require in the process of replication. It requires your parental DNA. So in this also you require a template DNA. Then you require two primers. Since here we are not forming a Y fork, and and here the two strands are completely denatured, not like this replication where your helicase will unzip the DNA and form a Y-like fork-shaped structure. So here actually you require two primers complementary to each of the strands of the parental template DNA. Then you also again require DNTPs, that is DATP, DGTP, DTTP and DCTP. So all the four nucleotides you actually require, and then the enzyme, of course, the Taq polymerase for the extension of the chains or for the polymerization of your template or the target DNA. Then you require a buffer where you normally contain these magnesium ions, which acts as a cofactor for the stack polymerization activity to occur. And then you put all these substituents in a thermal cycler, and you actually proceed through three different steps in the PCR. So the first one is the denaturation. As I discussed, that in the first step you need to completely unwind your DNA into single strand. Once your DNA is completely single stranded or completely unwinded, now these primers, which are complementary to each of these two strands, will come and try to anneal in these two sequences or in these two target DNA sequences. Now, once it got annealed, it needs to be extended. As I discussed, that the Taq polymerase has an optimum enzyme activity at 70 to 75 degrees Celsius. So this annealing, that at what temperature your primer will be annealed to the template DNA or the target DNA sequence, depends upon many factors such as the GC content and the TM of your primers. Okay, so on the basis of that, you need to optimize your annealing temperature. But finally, since the Taq polymerase works best in 70 to 75 degrees Celsius, we should increase the temperature to 72 degrees Celsius, where these new nucleotides, that is these DNTPs, will sit in the 3' end, and the extension of your chain will carry forward to give you an product or exponential product in this process, since you will carry out these three processes in cycles, and after 30 cycles you will get millions of copies of your target DNA. Okay, so if we go how these cycles are actually proceeding, that if you start with a single copy of your template DNA, then in the first cycle, you have only two copies of your DNA, which contains one strand of the template DNA and one of the new strand will be formed. But still now, your complete target sequence is not getting amplified. So that will be getting amplified only in the third cycle. But again, in the second cycle, you get 4 copies of your template DNA. In the third cycle, you get 8 copies of your template DNA. Similarly, in the 30th cycle, if you start with a single template, then you end up to 10 to the power 9 copies. So it seems that there is an exponential increase into of your template DNA copies as we proceed through the different cycles, and the exponential increase is in the 2 to the power n form. So I have also provided this YouTube link. So if someone of you are interested more to know about PCR or try to brush up your concepts about PCR, then you can actually look into this video for more greater concepts. So now if we again go to this question, and now let us read it carefully, since we have now a basic idea about this PCR amplification process. So what they have said, that they have given the total 1.2 kilo base pair DNA fragment was used as a template. So they have given the length of the template, that is 1.2 kilo base pair for the PCR amplification, and here instead of two primers there are four primers which are being provided in the question, that is P1, P2, P3 and P4. And you can see that in which regions of your template DNA which has a length of 1.2 kilobase pair total length, so in which regions these primers are actually complementary and binding to this DNA fragment, you can see from this image. Okay, and the positions from which means that it starts from here. 1 is the first nucleotide and the P2 binds in the 300 nucleotide. So these lengths are also being provided in the question. Okay, so how much distance this P3 and P1, if they are actually binding to the template DNA, what will be the amplified product length? You can actually easily find out from by looking into this figure. So how we will be actually discussing in the solution part. Then what they are asking, the annealing position of primers in the template are indicated by numbers. So these annealing positions at which they are actually being annealed, these primers, they are actually indicated by numbers as I discussed. Primers P2 and P3 contain single base mismatches as indicated by filled triangles. So what is there in these two primers is that if there is a single nucleotide mismatch, means if there is a C present in this template DNA, here should be a G. So instead of the G, here it is a or T. Means there is a mismatch which is present, but that is a single base nucleotide mismatch has occurred. So that is being indicated by this triangle. Whereas, so that is for both the P3 and P2, it is being indicated by this triangle. Then what they are asking, PCR was performed using primer pair P1 and P3 in one vial. So mark that in one vial, they have done this PCR by using this template and they have used P1 and P3. And again in another vial, they have used P2 and P4 and carried out the PCR for in each of these two vials. The purified PCR products from the two vials. So after carrying out the PCR from the two vials, in one vial by using P1 and P3 as the primer, and in another vial by P2 using P2 and P4 as the primer. After the PCR is done, they have purified the PCR product, and from the two vials they were mixed and subjected to another round of PCR with primers P1 and P4. So the product of the PCR1 containing the primers P1 and P3 is mixed with the product of PCR2 which contains the primer P2 and P4, by where the amplification occurred by the use of P2 and P4. Now they are telling that in the mixture of these two products of these two PCR, another PCR was carried out by using the P1 and the P4 as the primer. So what will be the final PCR product length? So for this, you need to actually see what is happening in each of the PCRs that is been asked in the question. So that's why I have actually drawn for your ease what actually is happening in each of these PCR cycles. So in vial 1 or in the PCR 1 where you have used the P1 and P3 as your primer, what is going to happen is that P1 is going to sit here to its complementary strand and the P3 is going to sit here, and they will actually extend in their through their 3' region, and they will be forming these two strands. One will be your parental strand, whereas in another one there will be a single base pair mismatch. Okay, in another product. In vial 2, again we use P2 and P4 as our primer. Okay, so what will happen again? So for P2 there is a single base pair mismatch. So there will be again a single base pair mismatch as one of the product, and one of the another product is the parental DNA or the template DNA itself. So these two ones you got, on mixing the two vials, P1 binds to first nucleotide at the 3' end and P4 binds to 1200 nucleotide at the 3' end. Since the total length is the 1200, okay, 1.2 kilobase pair. So that's why the P1 will bind to the first and the 1200 nucleotide will be bind to the P4. Thus, only those products from vial 1 and vial 2 that have these nucleotides can serve as template for this PCR. Therefore, the final product that can be formed is 1.2 kilobase pair. So, there are two single base mutations already present from these two PCR, and if you carry out the PCR by using these two primers, you will get the 1200 kilo base pair length products only, but there will be a pair of single nucleotide, a single base mismatch. So the answer to this question will be 1.2 kilo base pairs, that is 1.2 kilo base pair with 2-point mutation. So the answer to this question will be answer B. For your reference, you can go through these two courses, that is the Experimental Biotechnology course by Professor Vishal Trivedi of IIT Guwahati and another NPTEL course of Genetic Engineering Theory and Application, again by Professor Vishal Trivedi of IIT Guwahati, for clearing your concepts in this molecular biology techniques part.